Hey everyone, this is FixerMed. Welcome back to my high yield biochemistry review series for the USMLE Step 1 NBME CBSE and NBME CAS examinations. This will be part 8 of a multi-part video series detailing biochemistry in a high yield but comprehensive manner. So without further ado, let's go ahead and get started on today's lesson. Today we're going to be talking about G-coupled protein receptors and the effects of agonism on them and what receptors agonistic agents act on. So let's go ahead and break these down one by one. Hopefully I can give you some good mnemonics that help you memorize these, since G-coupled protein receptors give a lot of students a lot of trouble because it's just mainly just memorization, being able to retain that information throughout your test day. So let's go through these one by one. In GQ agonism, the main effects observed are increased phospholipase C levels, IP3 levels, and diacylglycerol levels, known as DAG. The main receptors that are acted upon in GQ agonism are histamine 1, alpha 1, vasopressin 1, muscarinic 1, and muscarinic 3. The main mnemonic I use for this receptor is HAV 1 or 3 M&Ms, where the HAV stand for histamine 1, alpha 1, and vasopressin 1. And 1 or 3 M&M stands for muscarinic 1 or muscarinic 3. Moving on, I'm going to skip GS for now and move on to GI. The main effects of agonism at GI are inhibited adenyl cyclase, which decreases cyclic AMP levels. The main receptors that get acted upon in GI agonism are muscarinic 2, alpha 2, and dopamine 2. The main mnemonic I use for this one is MAD2S. MAD stands for muscarinic alpha dopamine, 2 stands for 2, since it's only muscarinic 2, alpha 2, dopamine 2 that get added upon, and the S is just there to show it's a plural. Great way for it to stick. Now for GS, I don't have a mnemonic, so for this one you just kind of have to memorize it. So for GS agonism, the main effects observed are activated adenyl cyclase, which increases cyclic AMP. So the reverse of GI, basically. And the receptors that get acted upon are beta 1, beta 2, dopamine 1, histamine 2, vasopressin 2. So I'm going to now give some examples of agonism on these receptors through different substrates. So for GQ, phenylephrine, an alpha 1 agonist, can increase IP3 levels. On GI, agonism there, metal dopa or clonidine alpha 2 agonists can decrease cyclic AMP. Haloperidol, a D2 blocker, can increase cyclic AMP there. For GS, isoproterenol, known as a beta 1 and 2 agonist, increases cyclic AMP. Propranolol, a beta 1 and 2 antagonist, decreases cyclic AMP. So my tips for this section are to use mnemonics like HAV 1 or 3 M&Ms and MAD2S for GQ and GI receptors respectively, and then remember specific examples like phenylephrine, metal dopa, clonidine, isoproterenol, and propanol to solidify your understanding. So I hope those examples helped. I have a nice summary slide here of everything I've said, so if you just want to write it down, rewrite it, understand it that way, feel free to do so. Yeah, this is an NBME application-based reasoning. It's just summary slide. So yeah, moving on to the actual NBME application-based reasoning question. We have a researcher that develops a toxin that inhibits the GTPase activity of GSG proteins. Which of the following is most likely to increase in a cell as a result of this toxin? So GS. All right, take a few more seconds. Moving on to the next question now. If you need more time, pause the video. Moving on. So we have GTPases that normally function to shut off G protein activity. Inhibiting GTPase activity leads to constitutive, aka continual increased activity of the G protein. G-alpha-S G proteins, when activated, stimulate adenyl cyclase, which increases cyclic AMP levels. Therefore, inhibition of G2Pase activity would result in prolonged activation of adenyl cyclase and increased cyclic AMP production. Therefore, the correct answer here is C. Cyclic AMP levels would be the most likely to increase in the cell as a result of the toxin. Let's go down and break down why the other answer choices are wrong. So we have phospholipase C activity. G-alpha S is not directly involved in activating phospholipase C. B, inositol I4-5 triphosphate, known as IP3 levels. G-alpha S is not directly involved in the production of IP3. If you remember from what I was talking about, GQ was involved in IP3 levels, not G-alpha S. Diacylglycerol levels. G-alpha S is not directly involved in the production of diacylglycerol. Once again, that was a GQ. Adenyl cyclase activity. This might be a tempting choice, but the question is asking about the cellular effect of inhibiting G2Pase activity, not directly inhibiting adenyl cyclase. So these are why the other answer choices are wrong. Let's move on to the next question. An investigator studying a vancomycin-resistant bacterial strain observes vancomycin sensitivity and a frequency of 1 per 200 cells after many generations of propagation. What is the mechanism for the loss of this resistance? I'll go ahead and give you guys a few more seconds. Moving on now. If you guys need more time, pause the video. If not, moving on. Antibiotic resistance genes are located on bacterial plasmids. Loss of the plasmid results in the loss of the resistance phenotype. Plasmids consist of 3 main components, including the antibiotic resistance gene. In this case, the loss of vancomycin resistance suggests the loss of the plasmid containing the antibiotic resistance gene. Therefore, the correct answer is C, plasmid loss. Let's go through why the other answer choices are wrong. Mutations in the bacterial chromosomal DNA. A. The question refers to the loss of resistance, which is more likely related to the disappearance of the plasmid carrying the resistance gene rather than mutations in chromosomal DNA. Therefore, A cannot be the right answer. B. Acquisition of a new antibiotic resistance gene. The scenario described involves the loss of resistance, not acquisition of new resistance. Therefore, B cannot be an answer. D. Overexpression of the antibiotic resistance gene. This is not consistent with the observed loss of resistance. Moving on. E. Changes in the bacterial cell membrane structure. The question doesn't suggest any involvement of changes in the bacterial cell membrane structure in the loss of resistance. Therefore, E cannot be the answer either, and the only answer that made sense here was C, plasmid loss, since that is where the antibiotic resistance genes are located. Lost my train of thought there for a second. My bad, guys. But yeah, I know this is a very tricky and confusing subject for most folks, but be sure not to skip around it. It's high yield from what I've seen with the past NBME exams that have been floating around. So don't skip out on this unit. It's easy points if you can get this stuff down. Don't go too much in the weeds, but have a baseline understanding so you can get easy questions right, like which one causes IP3, DAG, etc. Mainly keyword recognition for this unit. As always, guys, if you have any questions, comments, or concerns, please feel free to leave them down in the comment section below. As always, this is FixerMed signing off. Please be sure to have a great day. Goodbye, everyone.